First, I would like to thank the meeting organizer for the invitation. Today, I'm going to talk about BPAN mouse model. BPAN is a disease that damages the nervous system and gradually gets worse with age. It's a subtype of NBIA, characterized by deposition of iron in the basal ganglia, known as tiger eye. BPAN patients show static encephalopathy and psychomotor retardation in childhood, and develop sudden onset dystonia, Parkinsonism, and dementia in adulthood. Therapeutic interventions for NBIA are currently limited, and the mechanism underlying the pathogenesis of the disease remains largely unknown. Human genetic studies reveal that BPAN is caused by mutations in the gene WDR45. My lab has identified that the C. elegans WDR45 homologue, known as EPG6, acts as an essential autophagy gene. Like a home, cells constitutively produce waste materials such as damaged organelles and protein aggregates. Failure to clean up the waste materials will impair the normal function of cells. Autophagy is a cell cleaning and recycling process. Waste materials produced in the cell is wrapped up in a little sac called an autophagosome. The autophagosome transports the waste material to another sac called the lysosome, which functions as a refuse processing plant in a cell. After the fusion of autophagosomes with the lysosome, the waste is broken down and recycled. Thus, autophagy is essential for maintaining cellular homeostasis. In neurons, autophagy can collect waste materials produced in axon terminals and deliver to the lysosomes in the neuron bodies for degradation. Yes, autophagy is critical for maintaining neuron and axon homeostasis. Neurospecific depletion of genes essential for autophagosome formation, such as ATG7, leads to massive neuron loss and axon degeneration. Samples from BPAN patients display impaired autophagic activity, suggesting that the dysfunction of autophagy is important for BPAN pathogenesis. WDR45 has a closely related symbolin gene called WDR45B. Mutations in WDR45B cause another neurological disease, intellectual disability. The mechanism underlying the pathogenesis of ID remains unknown. To understand the role of WDR45 and WDR45B in neuronal function, we genetically engineered mice to construct WDR45 and WDR45B knockout mice, then characterize the phenotype of knockout mice and investigate the mechanism underlying the pathogenesis of the disease. We generated mice with a depletion of the WDR45 in the central nervous system. WDR45 knockout mice are born normally and show no obvious growth retardation. WDR45 knockout mice exhibit no evident motor dysfunctions such as tremor and dystonia. We performed rotor rod experiments to examine motor coordination of the knockout mice. In this assay, mice are tested for their ability to remain on the rotor rod at different rotation speed. Compared to their control lead mates, WDR45 knockout mice at 7 to 8 months show no significant difference, but WDR45 knockout mice at 11 to 13 months spend less time on the rotor rod when assessed at higher speed, indicating that aged WDR45 knockout mice show impaired motor coordination. The hallmark neurological defect associated with BPAN is intellectual impairment. We performed behavior tests to determine the learning and memory ability of WDR45 knockout mice. The Morris-Water maze test involves recognizing and remembering visual cues to locate a hidden platform, thus measuring learning and memory function. In this assay, a round tub is filled with opaque water and the platform is located in the center of one quadrant. Compared to controlled mice, WDF45 knockout mice show significantly prolonged latency to find the hidden platform, indicating a learning impairment. Without the platform, knockout mice spend less time in the target quadrant, suggesting impaired retention of spatial memory. In a field conditioning test, mice are placed in the chamber and receive conditional stimulus, a 70 dB sound, and then adverse stimulus, an electric foot shock. After training, compared to controls, WDF45 knockout mice show a significant decrease in freezing time when the mice are put in the same chamber in the contextual test. These assays indicate that WDF45 knockout mice show significant deficits in cognitive function. Axon swelling is a key pathological feature associated with BPAN. To investigate the neuron defect in WDF45 knockout mice, we performed histological analysis on brain sections of control and WDF45 knockout mice at 13 months. Compared to controls, neuron loss is not evident. However, axon swellings are scattered in various brain regions. For example, Purkinje cell axons in the deep cerebellar nuclear region, detected by anti-carbidin antibody, showed huge bulging structures in knockout mice, indicating Purkinje cell axon swellings. Swollen axons are formed progressively. Thus, WDF45 plays a critical function in axon homeostasis. The autophagic activity is compromised in patients with BPAN. We examined the well-characterized autophagy substrate P62, which accumulate after inhibition of autophagy. Compared to control mice, in some brain regions of WDF45 knockout mice, P62 positive aggregates accumulate in neurons. In summary, WDF45 knockout mice recapitulate hallmarks of BPAN, including cognitive impairment, defective axon homeostasis, and autophagy defects. We also generate WDF45B knockout mice. WDF45B knockout mice are born normally and show growth retardation from 20 days after birth. WDF45B knockout mice show impaired motor coordination. In the roller rod assay, WDF45B knockout mice stay on the roller rod for significantly less time than controls, and the difference becomes more dramatic with age. WDF45B knockout mice also show tremor and seizure, and the defect becomes more evident in aged mice. Since mutations in WDF45B cause intellectual impairment in humans, we performed behavior tests to determine the cognitive ability of WDF45B knockout mice. In the Morris-Water-Maze test, WDF45B knockout mice show longer latency to escape onto the hidden platform. WDF45B knockout mice stay for significantly less time than controls in the target quadrant, suggesting a memory defect. In the field conditioning test, WDF45B knockout mice show a significant decrease in freezing time in the contextual test compared to control mice. Thus, WDF45B knockout mice exhibit impairment of learning and memory. We performed histological analysis of various brain regions of WDF45B knockout mice. The cerebellum of knockout mice are normal in foliation at early stages, but exhibit evident atrophy in size from 8 months old. WDF45B knockout mice also show a progressive loss of Purkinje cells, which become evident from 3 months of age. WDF45B knockout mice exhibit extensive axon degeneration. Swollen axons accumulate in the DCN regions in the knockout mice from 1 month old. Electron microscopy analysis also shows accumulation of a large number of swollen and degenerated axons in the DCN. WDF45B knockout mice exhibit autophagy defects in the brain. P62 positive aggregates accumulate in various brain regions of knockout mice. In summary, WDR45B knockout mice develop neurodegeneration as characterized by axon swelling and behavior abnormalities and learning and memory impairment. We construct WDR45 and WDR45B double knockout mice. In contrast to single knockout mice that are born normally, the double knockout mice are born smaller and all mice die within 6 to 8 hours after birth. Neither WDR45 nor WDR45B single knockout mice show accumulation of P62 aggregates in brain regions at 1 day after birth. In the double knockout mice at postnatal day 1, however, P62 aggregates accumulate in many brain regions. Thus, WDR45 and WDR45B function redundantly in neural autophagy. To further understand the molecular mechanism of WDR45 and WDR45B in neuronal autophagy, we generate WDR45 and WDR45B knockout neural 2A cells, which is a mouse neuroblastoma cell line. Compared with control cells, levels of LC3 puncta, which label autophagic structures, and also P62 aggregates are dramatically increased in the double knockout cells. Therefore, depletion of both WDR45 and WDR45B causes dramatic autophagy defects. Autophagy is a multi-step process. It involves the formation of autophagosomes and the fusion of autophagosomes with lysosomes. We found that autophagosomes are still formed in WDR45 and WDR45B double knockout cells. However, the double knockout cells show a defect in autophagosome-lysosome fusion. For example, upon blockage of lysosomal degradation by chloroquine, LC3-labeled autophagosomal structures are detected inside LAMP1 labeled lysosomes in control cells, while the LC3 puncta in double knockout cells are largely separate from LAMP1 labeled lysosomes. Further study revealed that WDR45 is mainly localized on lysosomes and WDR45B on lysosomes. They function together to facilitate the function of another autophagy protein, ADG2A, to tether the autophagosomes and lysosomes for fusion. Therefore, in neuronal cells deficient in WDR45 and WDR45B, the waste materials are picked up by autophagosomes, but the autophagosomes could not engage with the lysosomes for unloading and recycling. This disruption stops them from working normally. Here is a summary. WDR45 and WDR45B knockout mice develop learning and memory defect, show accumulation of swollen axons. WDR45 and WDR45B function redundantly in neuronal autophagy and are important for autophagosome-lysosome fusion. The WDR45 and WDR45B knockout mice provide valuable models for revealing the disease pathogenesis of BPAN and ID respectively, for investigating the possible role of autophagy in axon maintenance, and also for preclinical testing and exploration of therapeutic interventions. Finally, I would like to thank people in my lab who did the work. Dr. Yan Zhang at UMass Medical School, and Docs Cui Ji, Hongyu Zhao, and Dongfang Li at IBP, Chinese Academy of Sciences. Our research on BPAN projects was supported by the Orphan Disease Center and also funding agencies in China. Thank you for your attention.